Friends, I am Mahima Laharia from Vijayanagar College Hospital. Today we are going to study the important topic, that is the vitamin B12, which is also known as our cobalamine. It is also known as the energy vitamin, as it is very important from all the vitamins of the B12, vitamin B, as there are 8 vitamins B, and we know that vitamins are very important part of our balanced diet. So now we will study this topic in the various headings. First we will see the introduction or the history of the vitamin B12, then the unique features of the vitamin B12, structure of the vitamin B12, as the structure consists of the 4 major component, that is our cobalt metal, chlorine ring, carbon chain and the ligand. Then the names of the different forms of the cobalamine, means according to the ligand, the names of the cobalamine changes. If cyanide is there, cyanocobalamine, hydroxycobalamine, aquacobalamine, methylcobalamine and so on. So the details is there. Chemistry of the cobalamines. The cobalt metal is present in the different oxidation state. We will see the details. Functions of the vitamin B12. So the 8th function I especially define about the coenzyme and the reactions of the 1,2 shift biomethylation, reduction of the ribonucleotides. So the 1,2 shift have the 2 important examples: the glutamic acid conversion to the methyl aspartic acid and the conversion of the methyl malonyl coenzyme to the succinyl coenzyme. And the biomethylation is explained with the example of the homocysteine to the methionine with the help of our methyl THA. Then the next important point is the sources of the vitamin B12 and the deficiency of the vitamin B12. So let us start our topic with the introduction. Vitamin B12 has the molecular formula C63H88CO N14O14P. It is also called as the red vitamin, as it is red crystalline in color. In 1948, anti-pernicious anemia factor was isolated, crystallized and named as vitamin B12 or cyanocobalamin. In 1964, Dorothy Hodgkin awarded the Nobel Prize for discovering the 3-dimensional structure with the help of the X-ray crystallography. So we can have a look on the structure of the vitamin B12 here. It is having this one cobalt mineral in the center in the +3 oxidation state, a chlorine ring and a organic molecule. In 1973, vitamin B12 was synthesized by the Woodward. Now let us have a look on the unique features of the vitamin B12. It is also known as cyanocobalamine. It is unique in that it is the only vitamin that contains an essential mineral cobalt in the +3 oxidation state. It is the only organometallic compound that occurs in nature. Other organometallic compounds synthesized in the lab. Then these vitamin B12 are synthesized by the anaerobic bacteria, and this cobalamine uses the macrocyclic ligand or ring chlorine in place of the porphyrin. Means if we see the structure of the vitamin B12, it uses the chlorine ring in the structure in place of the porphyrin ring, which is present in our hemoglobin and other important complex. And this vitamin is needed in very small quantity, in milligrams only. If we compare to the other vitamin B which are needed for the adults, it's needed in very small quantities, in milligrams only. And this is also can be termed as the energy vitamin, as it increases the energy when the vitamin B12 foods are intake. It is very sensitive to the pH and the light. It is stable to heat, and they are as water soluble vitamins. It is involved in the methylation of the mercury to generate the toxic chemicals like the methyl mercury ion. All these features make the vitamin B12 very special, and it is having a very complex structure as compared to the structures of the other vitamins. So now let us have a look on the structure of the vitamin B12 in details. See the structure of the vitamin B12 complex. As we see the structure, there are main 4 components. First is a cobalt metal ion, which is present in the +3 oxidation state here. Second is our macrocyclic ligand, that is our chlorine ring. So we can say this our chlorine ring is made up of 4 pyrrole rings, that is our A, B, C, D. And this our chlorine ring it resembles porphyrin ring, but it differs as a methylidine linkage CH is missing here between the adjacent pyrrole rings, A and D. So because of the missing of the CH group between the A and D, they also block the pi delocalization of the electron. So as the blocking of pi delocalization of the electron, it results to make the Corian ring flexible. As our Corian ring is more flexible in compared to our porphyrin ring, so it can adapt the various conformations, so they are very helpful for the biological activities. So first component is our cobalt metal ion, which is present in the +3 oxidation state here. Second component is our macrocyclic ligand, that is our Corian ring. Third component is our complex organic part. Complex organic part is made up of 3 components. First is our phosphate group, second is sugar molecule, third is our organic base. So we can see the complex organic part. This whole is our complex organic part here. We can just make it O here. So this is made up of a phosphate group, our sugar molecule, and a nitrogenous organic base, which is also known as our benzimidazole. Here it is our 2 methyl groups are present in this benzimidazole ring, so we can just name it as alpha 5,6 dimethyl benzimidazole. So first component is our cobalt ion, second is our macrocyclic ligand Corian ring, third important component is our complex organic part. Then the fourth important component of the vitamin B complex is our ligand, which is coordinated to the cobalt metal ion. So now let us see, this R can be our ligand also. So any ligand which is coordinated to the cobalt metal atom is our fourth important component. Now when we talk about the fourth component, so it's your ligand coordinated to the metal ion. We can say that the ligand can be your OH group, can be your H2O group, it can be your cyanide group, methyl group, or any 5-deoxyadenosine group. So the 4 important components of our vitamin B12 complex are: the first is our cobalt metal ion, second is our Corian ring, third is the organic part, which is made up of a phosphate group, a sugar molecule, and an organic base, and the fourth important component is our ligand, which I have represented here with the R or L. So now let us see the details. According to the presence of the various types of the ligands on the 6th octahedral site, the names of the vitamin B12 complexes can vary. Now when we talk about the fourth component, so it's your ligand coordinated to the metal ion. We can say that the ligand can be your OH group, can be your H2O group, it can be your cyanide group, methyl group, or any 5-deoxyadenosine group. So the 4 important components of our vitamin B12 complex are: the first is our cobalt metal ion, second is our Corian ring, third is the organic part, which is made up of a phosphate group, a sugar molecule, and an organic base, and the fourth important component is our ligand, which I have represented here with the R or L. So now let us see the details. According to the presence of the various types of the ligands on the 6th octahedral site, the names of the vitamin B12 complexes can vary. Let us see the names of the different forms of the cobalamine according to the group present on this position, means this is the 6th octahedral site. So we can say that the 4 nitrogen atoms of the pyrrole ring gives the forms the 4 coordinate bond. Other 5th bond is formed by the nitrogen of the benzimidazole ring, and the 6th position is substituted by either any ligand, or we can say that R group. So now let us see once again, the cobalt is coordinated with the 4 nitrogen of the pyrrole rings. 1 nitrogen is from the imidazole, and 1 bond is formed by our any ligand or R. So let us see how the names of the different forms of the cobalamine depends upon the position of on this R or R. So if our ligand is water molecule H2O, so the name will be aqua cobalamin, or we can also write it as a B12A. If it is OH group, ligand is our OH minus, then we can also write it as a hydroxy cobalamin or B12B. If it is cyanide ligand, the name will be cyanocobalamine, or simply we can write it as a vitamin B12. Then if it is methyl group, it is the methyl cobalamine. If it is your 5-deoxyadenosine, it can be written as a 5-deoxyadenosine cobalamine. and this is our vitamin B12 coenzyme. Here this acts as a coenzyme. So now let us talk about the chemistry of the cobalamins. Cobalt in the B12 can be in the variable oxidation state. It can either be in the +3 oxidation state, +2, or the +1 oxidation state. It depends upon the situations. So we can say that the reduction of +3 takes place to +2 and further to +1. And in the biological systems, these reductions are carried out by the NADH and the FADH2. Now we can say, if we consider our aqua cobalamine, means it is a vitamin B12 which has our water molecule as a ligand and the 6th position. So the name is aqua cobalamine. When it goes to the reduction, means when 3+ state accepts one more electron, it get converted to +2 state. It changed to B12R form, and it again accept one more electrons and it get converted to the CO+1 state. So we can say that in this diagram there are the 3 oxidation states of the cobalt. In the first B12A it is 3+ state, in B12R it is +2 oxidation state, and B12S is the +1 oxidation state. 3 of the oxidation state will have the different colors due to the different transitions. So we can say that our first 3+ oxidation state, we know that the cobalt has atomic number 27, and in the 27 atomic number, if it loses the 3 electrons, it will be having the 6 electrons in our T2G orbital. So they are all paired. So in this condition we can say that this is a low spin complex and the nature is diamagnetic. It is red in color. Now we can talk about our reduced state. It is brown in color and it is also low spin. Then our super reduced, we can say as for the super reduced condition, and it is observed to be green in the color. Now we can say that the different names are given according to the different oxidation states to the cobalamins. If it is in the 3+ oxidation state, then it is written in the bracket as Cobalamine and B12A H2O. A for the H2O ligand here we are using. Then if it is 2+ oxidation state, it is written as Cobalamine. V12R in the reduced form is for R. And similarly for +1 oxidation state, these are written as the Cobalamine. V12S as for the super reduced, means it further reduce one more, take one more electron, so it converted to the super reduced state. Now as this here, V12S is a very reactive species. So now let us see some of the important reactions of the V12S. See the reactions of our vitamin B12S CO first system, means when the cobalt is in the +1 oxidation state and it is reduced state, super reduced state. So it acts as a strong nucleophile. It participates in the oxidative addition reactions, and it is also acting as a powerful reducing agent as it convert our oxygen to the water molecule in the biological system. As we can say that the vitamin B12S occurs naturally and it is non-toxic also, and it is very helpful for the various reactions, and it is having a lot of catalytic potential. So now just to have a look on this, it undergoes the halide coupling. It can also undergo the ring opening. It add to the double bonds and alkene coupling also. Then dehalogenations, hydrogenation. It can undergo the transmethylation, cyclopropanation, and rearrangement. Means this many catalytic reactions can be performed by the vitamin B12S in the +1 oxidation state. So we can say it is very useful vitamin. And now let us see the reactions in the form. If you want to take the screenshot of this is a full form of our cobalamine only. It undergoes the halide coupling, ring opening, addition to the double bonds, alkene coupling, dehalogenation, hydrogenation, transmethylation, cyclopropanation, and the rearrangement catalysis. So just to see the reaction here, so vitamin V12S in the +1 oxidation state can react with our alkyl halide, and this group will be substituted here, and here RCOX means our acyl halide group, and it is substituted on our cobalt molecule. Here it is cyanide group, means when it reacts with our cyanohalogens, so our cyanide group is substituted here. Similarly here double alkenes molecule is substituted, alkene coupling we have, and here ring opening is taking place, and in this our azomethane molecule is reacting, so a methyl group is substituted on our cobalt part. So these are the various useful reaction or the various synthesis of the cobalamine derivatives with the help of V12S in the +1 oxidation state, and we can also say these are the in vitro synthesis of our various derivatives from the cobalamine derivatives. Functions of the vitamin B12. Vitamin B12 is vital for the blood formation and the healthy nervous system. It helps in the formation of the blood and to maintain the healthy nervous system. It helps in the synthesis of the DNA and RNA of our genetic material. It helps in the production of the choline and another vitamin B and the methionine and amino acids, which helps us to boost up the energy level. It helps in the formation of the RBCs along with the iron, folic acid, copper, protein, vitamin C and vitamin B6. Vitamin B12 is also required for the synthesis for the formation of the red blood cells in our body. It protects the myelin sheath and it helps to maintain the proper functioning between the nerves and transfer the messages. It is essential for the cell growth and the multiplication, means it is essential for the cell growth and the cell division. It is also helpful in the synthesis of the phospholipid. Then the most important function, they act as the biochemical function of the cobalamins is that they act as a coenzymes. So what are coenzyme? As the word itself is a co plus enzyme. So they combine with the enzymes to catalyze the various reactions, and we can define it as coenzymes are the organic non-protein compounds that binds with the enzyme to catalyze a reaction, means they attach with the different enzymes and they will help to catalyze a reaction. So we can say that vitamin B12 act as a coenzyme. They act as a prosthetic group in a different enzyme. Prosthetic group means a non-proteinous part. They will attach with the enzymes and they will catalyze the reaction. So the vitamin B12 coenzymes, if they are inactive enzymes, they are termed as the apoenzymes, and the active enzymes, they will combine with our vitamin B12 and they will catalyze the reactions. Now let us see the details of the coenzymes and the enzymes which functions to perform the biochemical reaction. Let us see the important B12 coenzymes. So the important vitamin B12 which act as a coenzymes are 5-deoxyadenosine cobalamine and the methyl cobalamine. As we know, we studied in the structure that the cobalt is attached with the 4 nitrogens of the pyrrole, 1 with the nitrogen of the benzimidazole, and the L group will be bearing. So if on this position 5-deoxyadenosine is there, so it is 5-deoxyadenosine cobalamine, and if the methyl group is there, so it is our methyl cobalamine. So these are the two important vitamin B12 which are acting as a coenzymes, means they will combine with the various enzymes and they will catalyze the various biochemical reactions. So let us see some important enzymes. Along with them, they will combine and they will they will catalyze the reactions. So the important enzymes are methyl malonyl coenzyme A mutase. Second is our alpha lysine mutase. Third important enzyme is the methionine synthetase. This is very important enzyme, and we will see in the detail this. Methyl transferase, ribonucleic reductase, and the important enzyme one more, glutamate mutase, and there can be many more. So these are the name of the enzymes: methyl malonyl coenzyme A mutase, alpha lysine mutase, methionine synthetase, methyl transferase, ribonucleic reductase, glutamate mutase. So these are the important enzymes which will combine with our vitamin B12, and they will give help in the they will catalyze many reactions. So now some important biochemical reactions will be described here. So first we can say that there are the 3 important reactions which we will talk. That first is isomerization, bioalkylation or the biomethylation, and third is the reduction of the ribonucleotide. So first reaction which I am going to explain is the isomerization. Isomerization, we can say this is the isomerase reaction through the 1,2 shift. So here in the 2 diagram you can easily see these are the 1,2 positions of the carbon, and the H group is then changed to the second group. So it is the simple 1,2 shift of the substituent groups in any compound. So this type of the reactions are they are also termed as the hydrogen transfer enzyme and the simplest reactions of this type is the isomerization of our glutamic acid to the methyl aspartic acid. So here you can see the R is present on the first group and it is shifted to the second group with the help of the enzyme glutamate mutase, and the R group is represented as a CH NH2COOH. So this is an example of the isomerization of the glutamic acid to the methyl aspartic acid with the help of the enzyme glutamate mutase, and here vitamin B12 is acting as a coenzyme. So next we will see the let us see the important B12 coenzymes. So the important vitamin B12 which act as a coenzymes are 5-deoxyadenosyl cobalamine and the methyl cobalamine. As we know, we studied in the structure that the cobalt is attached with the 4 nitrogens of the pyrrole, 1 with the nitrogen of the benzimidazole, and the L group will be bearing. So if on this position 5-deoxyadenosyl is there, so it is 5-deoxyadenosyl cobalamine, and if the methyl group is there, so it is our methyl cobalamine. So these are the two important vitamin B12 which are acting as a coenzymes, means they will combine with the various enzymes and they will catalyze the various biochemical reactions. So let us see some important enzymes. Along with them they will combine and they will they will catalyze the reactions. So the important enzymes are methyl malonyl coenzyme A mutase. Second is our alpha lysine mutase. Third important enzyme is the methionine synthetase. This is very important enzyme, and we will see in the detail this methyl transferase, ribonucleic reductase, and the important enzyme one more glutamate mutase, and there can be many more. So these are the name of the enzymes: methyl malonyl coenzyme A mutase, alpha lysine mutase, methionine synthetase, methyl transferase, ribonucleic reductase, glutamate mutase. So these are the important enzymes which will combine with our vitamin B12, and they will give help in the they will catalyze many reactions. So now some important biochemical reactions will be described here. So first we can see that there are the 3 important reactions which we will talk. That first is isomerization, bioalkylation or the biomethylation, and third is the reduction of the ribonucleotide. So first reaction which I am going to explain is the isomerization. Isomerization, we can say this is the isomerase reaction through the 1,2 shift. So here in the 2 diagram you can easily see these are the 1,2 positions of the carbon, and the H group is then changed to the second group. So it is the simple 1,2 shift of the substituent groups in any compound. So this type of the reactions are they are also termed as the hydrogen transfer enzyme and the simplest reactions of this type is the isomerization of our glutamic acid to the methyl aspartic acid. So here you can see the R is present on the first group and it is shifted to the second group with the help of the enzyme glutamate mutase, and the R group is represented as a CH NH2COOH. So this is an example of the isomerization of the glutamic acid to the methyl aspartic acid with the help of the enzyme glutamate mutase, and here vitamin B12 is acting as a coenzyme. So next we will see the next example of the 1,2 shift isomerization is the conversion of the methyl malonyl coenzyme to the succinyl coenzyme. Here our 5-deoxyadenosyl cobalamine will act as a cofactor, and the methyl malonyl coenzyme A mutase will act as an enzyme. So our methyl malonyl coenzyme A is converting to the succinyl coenzyme, and this whole reaction is taking place in the mitochondria. So now let us see this reaction. Here our methyl malonyl coenzyme A, where the group is on the first position and it shifts to the second position with the help of our coenzyme activities, and the enzyme is here methyl malonyl coenzyme A mutase. So this is an important example for the 1,2 shift or isomerization. Next we will see the biomethylation. Important example of the biomethylation is in the metal ion toxicology, means many bacteria have the capacity to methylate the heavy ions, and they will convert this our mercuric ions and they will combine with our methyl group and they will form our either here methyl cobalamine will act as a coenzyme and they will form CH3Hg plus. And when this compound, this chemical, this ion is taken up by the aquatic plants and animals, it harms them, and many fishes or planktons or zooplanktons died because of this consumption. And if this is consumed by the human beings also, it has caused a disaster in our 1953 in the Japan, that is the Minamata diseases. It is because of the biomethylations in the metals, and it causes the heavy environmental health problems. Now we can say the important another important biosynthesis of the acetate from the carbon dioxide also takes place with the help of our vitamin B12 coenzyme, that is our 1,2S. We have written as vitamin 12S, means it is our the oxidation state is the 1 here. So same our methyl tetrahydrofolate, it will react with our it will provide one methyl group to our this vitamin B12S form, and it will get converted to our third oxidation state, and it will convert the carbon dioxide to the acetate form. So this is also important, the biosynthesis of the acetate from the carbon dioxide with the help of the our methyl cobalamine complex coenzymes methyl cobalamine. Next important thing to note here, that methyl cobalamine releases the methyl group in the 3 forms depending upon the situation. First, it is releasing the CH3 minus in the example of our CH3 plus ion, our first form we have just seen above only. Then second, it also releases the methyl as a free radical. We have not studied this reaction. It is the synthesis of the chromium methyl complexes. Then the third form, it releases the methyl group in the form of carbocation in the synthesis of the methionine. We already studied this in the synthesis of the methionine from the histamine. Then next, third important reaction of our coenzymes was the reduction of the ribonucleotide. Here we can see that OH group on the ribose second carbon is converted to the deoxyribonucleotides. The OH group is replaced by the H, but this reaction detail is not given much, so just we can say that the reduction of the ribonucleotide is also catalyzed by the vitamin B12 coenzymes. So now let us see the sources of the vitamin B12. Sources of vitamin B12. The important sources of the vitamin B12 are animal protein foods, especially meat, fish like trouts, salmon, mackerel, crabs, oysters are also good source of the vitamin B12. Egg yolk is very good source of the vitamin B12. The milk products, especially the yogurt, is rich in vitamin B12. Organ meats, especially the liver, heart and the kidney, are the good source of the vitamin B12. Veg source is only legumes, I can say that pulses, because we have already know in the leguminous plants that there is a rhizobium bacteria, so some amount of the B12 source can be obtained from the legumes, pulses and the almonds. So these are the important sources of the vitamin B12. The deficiency of vitamin B12 causes the anemia, fatigue or the nerve damage. It also causes the pernicious anemia, which is in the beginning itself we told the vitamin B12 as an anti-pernicious anemia factor. So pernicious anemia is an inability to absorb the vitamin B12, and it is caused by the lack of the intrinsic factor, a chemical present in the stomach lining. So if this chemical is missing in our intestines, so the body is unable to absorb the vitamin B12 and it lacks and causes the diseases, that is our pernicious anemia. And we can treat it with the help of the injections of the vitamin B12, and the person is having the disease pernicious anemia can be treated with giving the good diet of the liver, as in the beginning itself is told the organ meat liver has a good source of the vitamin B12. And we next the deficiency of the vitamin B12 causes a decreased sensory perceptions, diminished reflex response, means it's a quick response आप नहीं दे सकते हैं. It also causes the weaknesses of the arms or the legs, हाथ पैर में थोड़ा सा weakness feel होना, and the memory loss. A short time memory loss can also be caused due to the deficiency of the vitamin B12. So we should take a healthy diet which should be rich in the vitamin B12 sources, so we can not have the deficiency of the vitamin B12. Thank you all, and I hope you understand the topic well. If you have any doubt, please ask in the comment box, and please share and subscribe my channel.